In 1990, neovascular age-related macular degeneration, NAMD, and diabetic retinopathy, DR, were the leading causes of vision loss and blindness in older and middle-aged Americans, respectively.
I finished training as a vitreoretinal fellow in 1991, and at that time, treatment for many retinal diseases was really limited or non-existent. Neovascular AMD caused 90% of AMD-related blindness. And to treat neovascular AMD, all we had was laser photocoagulation that cauterized the abnormal blood vessels, but damaged the neural retina and led to a scar formation and loss of vision. It was a treatment that was not something that doctors liked to do or that patients really appreciated.
In the 1950s, the development of proliferative diabetic retinopathy was actually a risk factor for mortality at that point in time. In the 1960s and 70s, about half our patients were going blind every year from the condition. Now, after the 1970s, the development of laser panretinal photocoagulation reduced the risk of blindness by about 50%, but still there was a great deal of visual loss, and the laser treatment itself had some side effects, mostly built into the fact that laser was destroying areas of retina. There were problems with peripheral vision and night vision. So the need was for both a more efficacious treatment that could save more sight for proliferative retinopathy, also particularly for diabetic macular edema, where the laser helped prevent getting worsening but didn't really improve it, and also for a therapy that wouldn't destroy the retina in order to have these benefits. So when anti-VEGF came along, not only were we no longer destroying areas of retina to have the benefit, but the beneficial aspects of it were about twice as much as the laser were, and the side effects and the risks were 3 times less. And so this brought about a very rapid change in our primary therapies for these conditions.
Beginning in 1991, the National Institutes of Health, NIH, funded physician scientists identified the principal cause of vision loss in these diseases, vascular endothelial growth factor, VEGF.
We all grew up in ophthalmology knowing that there was a mysterious factor X that drove abnormal blood vessels in eye diseases. And investigators had been searching for factor X for decades. With the discovery of VEGF in the 1980s, and the fact that it was both secreted and driven by hypoxia, we in our group were excited that this might actually be factor X. When we could then show that VEGF inhibition completely prevented neovascularization in animal models, we began to get even more excited. And with our small clinical study, and then the larger one by Lloyd Aiello and George King, showing an association between VEGF and abnormal blood vessels in the eye, including proliferative diabetic retinopathy, we knew we were really onto something.
The development of anti-VEGF for the eye is actually a fascinating story, and one of the aspects of it is that it was first identified as a factor in cancers. And by looking at some of the studies that come out for cancer, it became apparent that this molecule both caused endothelial cells and vessels to proliferate and also caused them to leak. And those were 2 fundamental characteristics of the complications in certain conditions, and particularly in diabetic retinopathy, the area that we worked in. And so the question became, could one factor be causing these 2 major problems that result in visual loss from diabetic retinopathy? And as we started to study this factor, we found very rapidly it had all the characteristics that had actually been proposed for such a factor nearly 50 years earlier. It caused vessels to grow, it caused them to leak, it was increased when there was not enough oxygen around, it was diffusible within the eye, and it was found in the eye, and particularly in these conditions. And it was in about 1994 or so when the first studies came out where we looked at VEGF inside the eye in relation to these different conditions, where we found that it was dramatically increased in things like diabetic retinopathy or central retinal vein occlusion or retinopathy of prematurity. And once we had that association, then it became very likely that if we could find a way to block this factor, we would have benefits. And although that took many years to show, down the road these therapies became remarkably effective, for the first time actually improving vision in some of these conditions where at best before you were trying to maintain vision, and then became really the first line standard of care around the globe for these eye diseases.
In 1998, NIH-funded scientists partnered with biotechnology companies to launch the first anti-VEGF trials in ophthalmology.
The initial results were incredibly dramatic. The treatment with the anti-VEGF inhibitors, once the studies were unmasked and one could see what the effect of the actual drug was compared to not using the drug, was dramatic improvement. Not only did it prevent the worsening, but there was dramatic benefit in terms of improvement of visual acuity in many of these conditions. And that had really not been seen before for the conditions that were being studied.
For a long time, I actually had studied a different growth factor. When I was a postdoc at Hopkins, there was a very well-known ophthalmologist there who studied eye diseases in babies called retinopathy of prematurity. And it turns out to be an application for which anti-VEGF is actually being used now. And while I was there, I tried to find an angiogenic factor from the eye, and I found FGF, not VEGF. So I worked on that for a while. And then I came back to Boston as a first instructor and then an assistant professor at Children's Hospital in the group of Judah Folkman. He had a big angiogenesis laboratory.
The clinical trials of the anti-VEGF drugs were supported by private industry, including iTech, Genentech, Roche, and Regeneron, to name a few. Post-approval, NIH funding was used for the comparative effectiveness trials of the various drugs, in particular for AMD, and that would be the CAT trial.
He also had Tony Adamus working there. Dave Scheimer was a graduate student with me, and Joan was a fellow at the time. And we were all sort of working together virtually across the river, because we were down the river, actually. We were at Children's. Joan was at Mass Eye and Ear, although Joan used to schlep from Mass Eye and Ear to come down to my lab meeting, our lab meetings where we discussed this. She was very dedicated. So it was sort of a totally, I wouldn't say spontaneous, but a meeting of the minds of people who were all asking the same question. And we were doing laboratory research on it. As I said, we were, Joan had the animal model. We would get tissue from that. Dave Scheimer would run a Northern. That's what we were doing at the time, show there was message. Then we would get the fluid out and be able to do an ELISA to show that the VEGF was there. And then Tony and Joan would be able to visualize it and see that the blood vessels were there. So it was really a collaboration. It was the classic, it takes a village. And then the actual demonstration of inhibition, Tony and Joan initiated a collaboration with Napoleon Ferrara at Genentech at the time, who had developed a neutralizing antibody, which was actually developed at that time for use for tumors. So the initial development of anti-VEGF was for tumors, the angiogenesis and the leakage. People were doing a lot of tumor work. In parallel, we were doing work in the eye, because actually the differences are not that big. I mean, tumors have excess blood vessel growth and are a problem because they grow uncontrollably and they're leaky. The eye has a problem because vessels are growing uncontrollably where they shouldn't be growing and they're leaky. And it turns out the mechanisms are quite similar. It's hypoxia, it's excess VEGF. So nature is very economical in her use of molecules. And so we learned from the tumor people, and then the tumor people learned from the research that we were doing in the eye. A lot of crossover.
Since the introduction of anti-vascular endothelial growth factor drugs, legal blindness from neovascular age-related macular degeneration and diabetic retinopathy around the world has been reduced by half.
It's had a huge impact on the treatment of patients, initially patients with wet AMD. It's been called a number of things, such as a miracle and paradigm shifting and revolutionary.
Anti-VEGF has been a game changer. Diseases with no treatment or with very limited treatment are now controlled and treated with anti-VEGF therapy. Millions of people are treated annually around the world with anti-VEGF drugs, and vision has been saved for countless people.
From the basic science viewpoint, it's also been paradigm shifting because it's wonderful and heartening and inspiring to be able to see research, basic research that's done that goes right from the bench to patients and then has such a dramatic impact.
The development of VEGF was really a successful partnership of federal funding, private enterprise, and philanthropy, and really all parts were essential. Federal funding really supported the basic science discovery of VEGF itself and some of the translational work that led to its development for treatment for patients.
NIH funding was crucial for the discovery and the early development of anti-VEGF therapies, particularly with regard to identifying VEGF as a key factor. It was NIH funding that helped discover VEGF as a key factor mediating cancers, and that was the insight that allowed many of us to think perhaps it would be worthwhile also in the eye. Once we had data that there was a likelihood that the VEGF in the eye could theoretically be mediating these conditions, it was NIH funding that allowed us to characterize that in detail, proving the fact that the VEGF existed in the eye, proving that it could have the effects of permeability and neovascularization, and starting to characterize how it worked, what its receptors were, what cells made it, and how that might be regulated.
For the kind of work that led to this, which is discovery work, which was, at one point me taking cow retinas and putting them in PBS to extract angiogenic factors. That was all funded by the NIH, as was the early work that we did in the laboratory. So that kind of discovery work requires federal funding because it's a leap. And companies will fund research, but they are more interested, biotech and pharma, they're much more interested in funding sort of the application of whatever discovery you've made. So when it comes to discovery, coming up with the hypotheses and doing the key experiments necessary to test the hypotheses, federal funding is essential because it doesn't come from really very few other sources will fund the kind of discovery work that's necessary to come up with these kind of observations that lead to this endpoint.